In this video, I want to give an overview of the cloning project that we're going to be doing in class. Remember that this project involves molecular cloning of the green fluorescent protein, or GFP. And I want to give you, right off the bat, a little bit of a rationale for why we are doing cloning and why people might want to clone a gene. And so let's start off by looking at a plasmid. So a plasmid is a circular piece of DNA, and often the definition of a plasmid is a small circular piece of DNA. They're often found in the wild inside of bacterial cells, and they often carry genes that these bacterial cells can transfer from one bacterium to another. And so, for example, if you find an antibiotic resistant bacteria, it often will have a plasmid inside of it that contains a gene that gives it resistance to an antibiotic like ampicillin resistance. And these plasmids can be passed from one bacterium to another, and they're separate from the genome or the chromosome of the bacteria. You can think of them as really small chromosomes with just maybe one or two genes, but they offer to the bacteria some sort of benefit in survival. Well, plasmids are also, in the lab, very useful constructs or very useful tools to be able to manipulate DNA and use DNA and study genes. For example, green fluorescent protein. And so often these plasmids are also referred to, so this is a plasmid here, a circular piece of DNA that we have co-opted from bacteria to use as a tool, but they're often also referred to as vectors because they will often use them as kind of a carrying case for a gene that we're interested in studying. So a vector is basically a term that refers to the ability of the plasmid to hold on to this gene and then deliver it into something that we're interested in studying. For example, maybe we want to know what this gene's role is in a cell, and so we can use this plasmid as a vector to deliver this gene into a cell. And then once in the cell, that could be expressed. Now you know from your previous classes that in order to express a gene in a cell and be able to learn about its function, the cell needs to be able to recognize the DNA and recognize that there is a gene there and do the processes of transcription to form the mRNA, and translation to create the protein from the mRNA. And you've also learned in previous classes that in order for these processes to happen, transcription and translation, you have to have certain elements in the DNA to indicate to the cell or to trigger the cell to do these processes of transcription and translation. So let's take a look at this gene. This is just some generic gene that we have, and I'm not going to draw the double-stranded DNA here. We can just pretend this is double-stranded. And you know that this contains the code for a protein. It contains the sequences of codons, or 3 bases, in a particular order that read out a particular sequence of amino acids when it's translated. You also remember that every gene has a start codon, and that's always going to be an ATG, and that indicates to this machinery of transcription, where do we start sorry, that indicates to the machinery of translation, so to the ribosome, that indicates where do we start translating the protein. We also have at the other end of the gene a stop codon, and that indicates to the ribosome we should stop translation here. So these two things, the start and the stop codons, regulate translation. And so when the ribosome is reading the mRNA, and in the mRNA, by the way, it would be AUG and UAG, and also remember that, so this would be the DNA, ATG and TAG, but when it's transcribed into mRNA, it would be AUG and UAG. Also keep in mind, I've just written one of many, well, there's actually 4 different possible stop codons. I can't remember all of them right off the bat right now, but I know another one is TAA, but there are about, I think, 4 stop codons. But basically what happens when the mRNA is read by the ribosome recognizes an AUG and it starts translation, and then it reads through the mRNA and adds amino acids depending on what the codons are that are in this mRNA, and then when it gets to the stop codon, the translation stops and the protein is released from the ribosome, and then it folds on its own and starts functioning as a protein. So if we're going to have functional protein produced from our gene, when we put in this plasmid into the cell, then that gene will need to have a start codon and a stop codon so that that protein is translated correctly. But there are other things that the cell needs, or that the gene needs, in order for that to happen correctly. Another thing that this gene needs to have upstream of the start codon is an RBS, a ribosomal binding sequence. The RBS is a sequence of DNA that the ribosome recognizes. It's not the start codon, but it's a sequence upstream of the start codon. And when I say upstream, I mean in the 5' direction. If this was the 5' end of the DNA and this is the 3' end. Upstream basically means before, if we're reading this from left to right. So there needs to be a ribosomal binding sequence that the ribosome can recognize and bind to. Now this is going to be transcribed into the mRNA, so the mRNA will also have a ribosomal binding sequence, as will the gene, but of course the ribosomal binding sequence in the mRNA is the one that's going to be recognized by the ribosome, not the RBS in the gene. But you've got this RBS that's upstream of your start codon, and this is a sequence necessary for the ribosome to be able to recognize the mRNA and attach to it. Okay, so those are 2 important things that the gene must have in order to be translated. But what about transcription? What kind of sequences do we need for transcription to happen? Well, transcription doesn't happen just with the gene itself. You need more. You can't just get transcription if you have just this gene sequence. This CDS coding sequence is often referred to, we often refer to the coding sequence, the CDS, as the gene sequence proper, starting with the start codon and ending with the stop codon. But you can't just put a CDS into a cell and hope that you get a protein out of it. You also have to have the ribosomal binding sequence. And to get transcription, you have to have some other elements on the DNA that are even further upstream. And one of the most important of those is called the promoter region. And you have probably learned about promoters already. But a promoter is a sequence on the DNA that allows for transcription to happen. It promotes transcription, thus the word promoter. And one of the main things that it does is it binds transcription factors And one of the most important of those transcription factors is RNA polymerase. RNA polymerase II to be specific. This is necessary for transcription to happen because, of course, RNA polymerase is what creates the mRNA, and so it, together with other transcription factors, bind to the promoter region and transcribe the DNA into the mRNA. You might also have upstream, for even further upstream, a sequence called an enhancer. And this is a region that interacts, this is a region of the DNA that interacts with the promoter and can actually enhance, or in other words, make transcription more likely, more frequently, make it occur more frequently, so that you have more mRNA produced. Now, more mRNA, what does that mean for the cell? You have more mRNA, that's going to mean more protein, because the more transcription you have, the more translation you have, because the more transcripts you'll have in the cell. So enhancers can increase the number of mRNAs that are produced. All right, let's look at what else you might have in the DNA that helps control transcription. There are other sequences that you find at the end of the gene, at the downstream from the CDS, that are also regulatory sequences, and one of them is called a poly-A signal. Remember that mRNAs, typically you have the sequence of the gene, and then typically you have a poly-A tail at the end of your mRNA. This is not found in the gene, it's not found in the DNA, it's added after transcription happens. But there is a signal downstream of the coding sequence that must be there in order for the polyadenylation for these poly-A's, for these multiple adenosines to be added. And though that poly-A tail is going to be necessary to regulate the lifetime of the mRNA, in other words, to protect it from being degraded in the cell, it keeps the mRNA intact until it's translated. So that poly-A signal is important, and so that poly-A tail is important. So therefore you need a poly-A signal that can trigger the cell to add that poly-A tail. So there needs to be a signal down here to do that. So that's all the regulatory elements that I'll talk about right now. But I want you to know, basically I want you to see that when we want to make a protein in a cell, there's lots of elements that need to be on the gene in addition to, beyond just the coding sequence, beyond just the codons of the gene. So what does that mean for us in cloning? Well, if we think about studying some sort of gene that we've got in a plasmid here, we need to make sure that when we put this gene into the cell, that it is surrounded by these regulatory regions that we just talked about, both upstream and downstream. Otherwise, that gene won't be expressed properly in the cell. And there's another complication too, which is that there are multiple kinds of ways to regulate the gene depending on what cell you want to put it in. So, for example, we may be wanting to study this gene in a mammalian cell, or we may be wanting to study this gene in a bacterial cell. And depending on whether we want to study it in a mammalian cell or a bacterial cell, the promoter we put, or the promoter that we engineer to be upstream of the coding sequence, needs to be one that's recognized by that type of cell. Not all cells will recognize and be able to utilize all different types of promoters. Mammalian cells have certain types of transcription factors that are mammalian specific, and bacterial cells have other transcription factors that are bacterial specific. And so if I want to express this gene in a bacterial cell, I need to have a bacterial promoter so that its transcription factors recognize that promoter. And for mammalian, the same thing. So there are many different types of promoters, and depending on what cell I want to express and study this gene in, I need to have the proper promoter here upstream. So what we're going to do in this lab is take a gene that's in the GFP gene that's in a plasmid called PGFP, and we're going to move it to another plasmid called PQE31. And the reason we're going to do that is because PQE31 has a promoter that PGFP doesn't have that we want this GFP to be controlled by. And so we're going to move this GFP gene to this plasmid so that we can, which contains already this promoter that's going to give us special control over the expression of GFP. And this plasmid, PGFP, that this gene is currently in, does not have that promoter. It does have a promoter, but it's not the one we want. What I'd like to do now is show you a map of this PGFP plasmid and then show it side by side with the PQE31 to show you the difference in the promoters. And this is basically the rationale or the reason why we're doing this cloning experiment to move this GFP from this plasmid, which doesn't have the promoter we want, to this plasmid, which does. So what you're looking at here is something called a plasmid map. I won't explain all the elements of this plasmid map right now, but you'll understand most of them by the time we're done. The first thing I want you to notice is the GFP gene. It lies right there. And this gray area basically indicates the sequence of the GFP gene. And notice that it's in the shape of an arrow. The reason that they do that is that they show you how this gene is read. Where is the start codon? Where is the stop codon? When you see an arrow like this, that means that the start codon will be somewhere up here and the stop codon will be somewhere down here. And so that gene is read in that direction. So this is the GFP gene. And its start codon is up here near something called the PLAC. This is a promoter. And it's a bacterial promoter. It's a promoter that bacteria use for another gene, typically in bacteria, in a normal wild-type bacteria. This promoter would be found upstream of a gene called lactase, or LAC-Z. And we have co-opted this promoter and put it into this plasmid here that we've engineered, and we've put this GFP gene downstream of that. So what this means is if we put this PGFP plasmid into bacteria, if we transform it into bacteria, which is a term, transformation is a term when you put exogenous or foreign DNA into a bacteria, when we put or transform this PGFP into bacteria, they will express it because there is a promoter. The other things that aren't shown here, but that are in the plasmid, but the map's not showing it, are the ribosomal binding site, and down here at the 3' end, it's not shown here, but we would find what we call the 3' regulatory sequences. And this may include the poly A signal. Now the reason I say it may include, and I'm not for sure if it does, is because some bacteria don't require the poly A signal in the transcription of their mRNAs. But because it has that promoter, if you put this plasmid into bacterial cells, they would produce GFP. Where does GFP come from? Remember, it comes from jellyfish. So if we put this into bacteria, we are creating what we'd call a transgenic organism, or a genetically modified organism. Another way you could say it is we are putting recombinant DNA into a bacterium. Even though this is GFP that's not normally expressed or found in bacteria, they will produce it. And GFP is typically non-toxic unless it's produced in really large amounts. And so these bacteria would probably be relatively healthy. They would just be fluorescent green. Okay, I want to show, before I show the other plasmid and give you a comparison between these two plasmids, I also wanted to show you a couple other elements of this map. So that, again, this is called a plasmid map. And right in the middle of it, we see what the name of the plasmid is, and we also see what size it is. So 3.3 kb is around 3,300 base pairs. So this whole circle is about 3,300 base pairs long. There's another gene. There's actually two genes on this plasmid. The other one is called an ampicillin resistance gene. And this gene contains the codons that will produce the gene or the protein beta-lactamase. Now what this protein will do is it will give to the bacteria a special power. It will give to the bacteria that contain this plasmid resistance to the antibiotic ampicillin, or AMP for short. Ampicillin is a cousin of penicillin, and normally penicillin and ampicillin will prevent bacterial growth. But if the bacteria has this plasmid inside of it, it will be resistant to that antibiotic, and the bacteria will be able to grow. And so this plasmid has that. Now, why does it have that? Well, if you think about being able to study this gene, this GFP gene in bacteria, if you want to study this gene or you want to produce this gene in bacteria, then you need to be able to grow bacteria that have your PGFP plasmid and prevent the growth of bacteria that don't have the PGFP plasmid. And the way you do that is you kill off these bacteria by adding ampicillin to all of your bacteria. And when you do that, cells that don't have the ampicillin resistance gene, because they don't have the plasmid, won't survive, and only the bacterial cells that do have the plasmid will live. This is called artificial selection, well, as opposed to natural selection, right? We are artificially selecting the bacteria that we want to survive and killing off the ones we don't want to survive. And this gene here, this ampicillin resistance gene, is called a selective marker. It basically allows us to select for bacteria that have the plasmid. Now, if they have the ampicillin resistance gene, they will by default also have the GFP gene because they're on the same plasmid. The last thing I want to show you is this PUC ORI. This is what we call an origin of replication. And this is very important for a plasmid to have, because if you don't have an origin of replication, when you are put into the bacterial cells and the bacterium divide, they won't be able to replicate the plasmid and give it to their daughter cells. So what we want when this bacterium divides into daughter cells is for this plasmid that's in the mother cell to be replicated into two plasmids, and each of these plasmids end up in the new daughter cells. And this origin of replication then triggers the bacterial cell to replicate the plasmid, otherwise it wouldn't. So this is crucial. You have to have this in the plasmid. You don't have to have ampicillin resistance gene in your plasmid. Bacteria can function just fine without an ampicillin resistance gene. It's just that if you try to select for the bacteria that have the plasmid, you'd have a really hard time doing that because you wouldn't be able to control you wouldn't be able to distinguish between the cells that do and don't have the plasmid because there's no difference in terms of their resistance to ampicillin. Okay, now let's look at the plasmid that we are going to be putting the GFP into. So this is our target plasmid, or it's going to be our target vector. There are actually three different versions of this vector called PQE30, PQE31, and PQE32, but the one we're going to be working with in the lab is called PQE31. They're all very similar to each other. There's only slight differences that we don't have time to get into in this video, but maybe in class or in another video we might talk about those. But essentially, long story short, the PQE30, 31, and 32 differ by just two bases. So PQE31 has an A and a C that PQE30 doesn't have. PQE31 has a G that neither of the others have. Here's the plasmid. Now let's start with things that are in common with the PGFP. So it's almost about it's very close to the same size, the whole plasmid, as PGFP. Again, this is about 3,400 base pairs, and it has an origin of replication also, and it also has an ampicillin resistance gene, which is the same one that we see in PGFP. The big difference that we see with PQE31 and PGFP is right up here in this region. Now, this region is just shown here by a white arrow, but what they've done is zoomed in on this region to show us what different elements are in this area. And again, this region of the plasmid would be read in this direction from the left to the right. Notice that it's different. It's in a different orientation from this ampicillin resistance gene, which is actually read from the right to the left. But one of the big elements that I want us to understand is right here. This is different. This part of the plasmid is different from the PGFP plasmid. The PGFP plasmid does not have this. So there are two different parts here, the PT5 and the LAC-O. Let's talk about the PT5 first. This is a promoter also. This PT5 promoter is different from the PLAC promoter. It's what we consider a stronger promoter. Now, what do I mean when I say stronger promoter? When this is put into cells, specifically bacterial cells, this promoter drives expression of the downstream gene more, so that you get more mRNA in the end. So when we say a stronger promoter, it's basically more active, and it will produce more mRNA, and therefore you'll get more protein. So if you want to study this promoter and you want to make lots of protein in the cell so that you can see what this protein does, then you want to use a strong promoter that will activate transcription more readily, more often, more frequently, and you'll get more mRNA and therefore more protein. So that's going to be good for us because we want to make high levels of GFP. Our ultimate goal is to transfer the GFP gene from PGFP to PQE31, but then also produce the GFP in the bacteria and collect it. And so we want those bacteria to become little GFP factories and produce a lot of GFP for us. And so we want this promoter, we want this GFP to be driven by a strong promoter. Now here's the thing, remember when I said GFP is typically non-toxic for cells? It's true in that GFP itself doesn't affect the other proteins in the cell. But there can be a problem if you make too much GFP. And with this PT5 promoter, you have the possibility of creating a lot of GFP in the cell. And this does a couple things. This causes the cell a couple of problems. First of all, if you have a lot of this protein filling up the cell, it may kind of clog up the works of the cell. If you think about all this GFP inside the cell, it may prevent the smooth workings of the cell, transport of things from one part of the cell to the other. The GFP may even start to clump together because there's so much of it. And so it can be a problem if you make too much of a protein, even though the protein itself isn't toxic per se. If you have a lot of something, even if you drink a lot of water, right, you can die. Not that water's poisonous, but just because it could cause other problems if there's too much of it. The other problem that producing a lot of GFP causes is that it puts strain on the cell, because the cell's machinery is not built to produce large amounts of GFP. And so it's taxing on the cell, it's taxing on its resources to be forced to produce a lot of GFP. So if I add GFP to this plasmid and then transform this plasmid into bacterial cells, they will be forced basically to produce a lot of GFP, which will tax their system, and it'll make them unhappy and unhealthy. So we've got competing demands here. On the one hand, we as scientists want to produce a lot of this GFP so that we can collect it and study it. On the other hand, the cells become unhealthy when we produce a lot of GFP. So here's the solution. We're going to use this part of the plasmid to be able to control when and how much the GFP is turned on or produced. This is called a lac operon. We'll discuss this much more in another video when we get to the day when we're expressing protein. But I just want you to take away the message I want you to take away today is that this is a DNA sequence on the plasmid that allows us to prevent the promoter from being activated until we say so. And we're going to say so by adding a particular chemical to the cells. And when we add that chemical, it's called lactose, it's actually a sugar, it will allow the promoter to turn on and produce a lot of GFP. But if we don't add that sugar, when lactose is not present, this lac operon DNA sequence prevents RNA polymerase, which binds here to the promoter, prevents it from being able to transcribe the mRNA and produce GFP. And so what we've created here is a strong promoter, but that is controlled by this operon so that we don't always have GFP being produced. We as scientists can control when it's produced. And so what we can do is allow the cells to grow without the constraint of having to produce a lot of GFP. We can let them grow and multiply and divide until they fill up the flask so that we have a lot of bacteria and they're all very happy and healthy. And then we can add the lactose. Once we do that, the cells will immediately start producing GFP, and they will be unhappy at that point, but there will be so many of them that there will be a lot of unhappy cells, but they will produce in mass a lot of GFP protein. So we've got this promoter, this PT5, combined with the lac operon that allows us to produce a lot of GFP, but only when we say so, by adding the lactose. That means that this whole promoter is something we can turn on or induce when we want, and therefore we call it an inducible promoter. And the PGFP plasmid does not have that. That's not an inducible promoter. This one is. And so that's why we want to put the GFP remove it from the pGFP, and add it to the PQE31. Notice that this plasmid also has that ribosomal binding sequence that I talked about, and it has some other things that I'm not going to talk about right now because it would overwhelm you with all that information. But you will know, and you'll be expected to know what these other things are later. But they will all make sense in due time. For now, I just want you to see what the difference is between the pGFP and the PQE31 and why we might be moving it from why we want to move that GFP from one plasmid to another. It's so that we can control the expression of this GFP, turn it on when we want to, have this inducible promoter.